It has been well established that the structures of many proteins are stabilized through covalent disulfide linkages. In recent work, this bond has been classified as a post-translational modification. Thus, it's important to be able to identify cysteine-stabilized multimeric complexes in living cells using a method that's quick and simple. The main advantage of this technique is that the results can be obtained and interpreted quickly, easily, and with minimum costs.
To begin, grow U2OS cells in a 6 square centimeter dish in minimum essential medium high glucose, supplemented with 10% FPS and 1% sodium pyruvate, at 37 degrees Celsius in 5% carbon dioxide. Make a fresh stock of 10 millimolar iodoacetamide just prior to use. After cells reach 50 to 60% confluency, add 0.1 millimolar final concentration iodoacetamide directly to the cell culture media. Gently rock the dish at room temperature for 2 minutes. Next, aspirate the media from the cells and wash the cells with 5 milliliters of cold PBS 3 times. Aspirate the final PBS wash solution and add 1 milliliter of cold PBS. Scrape the cells off the bottom of the dish using a cell scraper. Using a 1 milliliter pipette, draw up the PBS and cell suspension and dispense all the liquid into a 1.5 milliliter microcentrifuge tube. Spin the cells at 7500 times G and 4 degrees Celsius for 3 minutes. Aspirate the PBS, leaving the cell pellet behind. The cell pellet can be stored at -80 degrees Celsius